in relation to that question about pollinators and plants, this is from a study. I took this figure for an illustration out of a study we recently did, which is comparing the distribution of butterflies in North America and plants. And so if you want more information about how you would look at ecological interactions through these methods, we could talk more about that. But this nice figure came out of that paper, which was just a nice illustration of, looking at the distribution of organisms with a phylogeny, and then looking at the measures I'm going to talk about here in a minute to explain exactly what these mean, and then looking at the patterns that you get. And they're pretty spectacular. But first, and apologies if you already know all about phylogenies, but many people have trouble with this aspect of it. And so I just want to be completely inclusive by starting from the ground up. We say we want to know the phylogenetic relationships of organisms. And so how the heck do you do that? You can't see it directly. When I go for a hike and I go walk on Mount Diablo, I can see all these different plants, and I can see they have different characteristics, but you can't directly see how they're related to each other. That's not something that's on their sleeve. So how do we do that? One way to think of it as is if you had a big oak tree that was completely buried in a sand dune, and only the very twigs of the branch were sticking out of the sand dune, could you reconstruct the order of branching of those tips of the tree with only the data from the tips of the tree, without being able to undo the sand dune and see how they're actually connected? And the answer is, you can. We can actually reconstruct history without being able to directly see it. And the way we do it in this kind of a system, which is the relationships are branching with modification, is this idea of historical markers that have changed during a period of time and serve as markers. And so I'm going to explain what that is. You can do it with languages. It's how you figure out that French and Spanish are more closely related than either is to English. You sort of are looking at language as though it was a tree, which to a large extent it is. We inherit languages as people move around the globe. And then languages evolve, and you can make trees of languages and you can make trees of biology with only the data that you can see in the present day without actually being able to step back in the past. And that's this concept of a historical marker, which I'm going to have in the next slide. I'm going to show you what I mean by that. We need to find something which is different in some recognizable way and that has survived in its form to the present. So here it is in a nutshell. We have 5 things here. This could be 5 different trees that we observed on our hike. Real trees now. So we saw, a couple of pines, and then we saw an oak. Let's say those are the 2 pines. We saw a couple of pines, we saw an oak, and we saw a madrone. And we want to know, well, of the 4 things that I saw, which 2 of them are more closely related to each other? What we're looking for is some kind of a marker which changed its state, changed its condition in the past, in a period of shared ancestry of 2 of them that's exclusive of the others. So if this was language evolution, you're looking for some change in a grammatical structure or a word. The methods have been used to reconstruct the history of medieval texts that were copied by scribes one after another. And you can tell that the manuscript in this cathedral and this cathedral are related because a scribe made the same mistake in them. So you're just looking for anything that changed its condition, which then could be taken as at least one little piece of evidence that those 2 are more closely related. So each of these markers is just one little piece of evidence. The data sets we're eventually going to use are going to be enormous. They're going to have thousands of such kinds of evidence, and we're going to contrast them with each other. But in principle, it's really simple. Each little piece of evidence is just one putative change that happened, one mistake in a manuscript, one change in a word in language, or perhaps one change in the genome in biology. But it's a very general concept. Anything which has these kind of branching sorts of relationships will hopefully have these little bits of data There'll be some characters that change here. There'll be some that change here. And by looking at all of them, I hope that I can sort out what the relationships of these tips are. Okay. So to make it a little bit more complicated, just a few more tips. So here are 5 tips of the tree here. And a little more complicated because there were 2 changes in whatever this marker was. It started out in this open circle, and then it went to this gray circle, and then it went to this black circle. And when it changed here, it's a little piece of evidence for that branch. Oops, sorry. And when it changed here, it's a little bit of evidence for that branch. So this could be, if it was a genetic character, this could be an A at a site in the genome, and it changed to a C, and then the C changed to a T, that sort of thing. Evolution keeps happening, traits keep changing. And then some terms that we'll be using, I've already used monophyletic group. When we say monophyletic group, we mean a group that shared a common ancestor and all the descendants are in that group. Whereas a paraphyletic group, you'll hear that term, that is a group which did have a common ancestor, but does not include all the descendants of that ancestor. It's leaving these guys out. So at the big scale, an example of a monophyletic group would be the birds, and an example of a paraphyletic group would be the dinosaurs, because birds are nested in dinosaurs, but they're more closely related to some, including the group that includes the Tyrannosaurus rex, than they are to the rest of the dinosaurs. So we have in taxonomy, we have many groups that are monophyletic and many that are paraphyletic. And the modern trend has been to get rid of the paraphyletic groups and try to name only monophyletic groups. So that's what we mean. We say a monophyletic group is like one branch of the tree of life. You could grab a hold of the tree of life and break it once, and you'd be holding a monophyletic group. And then terms that you'll hear of apomorphy and plesiomorphy, that's the posterior state, and plesiomorphy is the prior state. So this is changing from one kind of genetic state to another, or one language state to another, so forth. So those are some of the terms that we're using here. And I'm gonna, here's a few more terms, and you guys probably know these, but just in case. So homology is one of these markers, which is shared by 2 lineages because of descent from an ancestor that had the feature. So homology is not just any similarity, it's a particular kind of similarity that's due to common ancestry. The transformation is what we call that evolutionary change from the prior state, the plesiomorphy, to the posterior state. Then a couple of things happen to lineages. In the Darwinian diagram, I showed how sometimes one lineage splits into 2 lineages. And the whole study of that is a very interesting part of biology. Sometimes it is one individual, or maybe a small group of individuals gets blown over to an island and forms its own new lineage, like in Hawaii, let's say. So that's a case of divergence. Then sometimes in evolution, you have reticulation, which is the opposite. This comes from the Greek for a network. So this is when you had 2 lineages, but now they blend back into one. And that happens too. We have to take that into account. And then I already explained what monophyly means. It's a group that contains all and only descendants of a common ancestor. So examples of monophyletic groups are birds and mammals, whereas examples of paraphyletic groups would be dinosaurs or reptiles. Just to illustrate the concept of homology, to be sure that we're clear on it, if we compare the 4 limbs of a human, a dog, a bird, and a whale, we find exactly the same elements are present in each, but they've been modified in each. So the bone in red here is homologous in the sense that I just mentioned. It's shared by common ancestry with all of these animals, but of course it has then been modified again in especially in the whale. Okay, so that's this idea that you can keep changing in a homology. It isn't fixed, but yet you can spot these. So I think you can think of many examples of homology that are morphological or anatomical, such as these. But then you also have homology at the DNA level, and what's called a sequence alignment, and this is really important, so I'm going to take a second on it, is when we sequence a gene from an individual organism, that's an item of data. And then we can sequence a gene from a different organism. And if they're homologous, meaning they have common ancestry and they're similar enough, we can align them. And what's called alignment in DNA is where you're taking the sequence from taxon 1 there, and you're laying it up against taxon 2, and you're trying to match them as closely as you can, and then look for the places where they are different. There is software for this, and it's, the field of bioinformatics is an absolutely enormous field these days, lots of employment opportunities, because this is very important in medicine and many aspects of human health. So, when you're trying to find a problem in a gene, for example, that some individuals have and some individuals don't, then you have to align the gene from the different individuals, and you have to look for these kind of places, which are called single nucleotide polymorphisms, or SNPs. And you look for those, and you see if maybe people that have a certain condition are more likely to have a certain SNP than the people that don't have it. And that helps to direct you to what might be the causal agent for whatever this ailment is. And so that's in taxonomy. What we do is we line these things up next to each other. And can everybody see how these mostly align? But in highlight here are places where they don't match. They match everywhere else, so they seem to align well. But it looks like taxon 3 has this sequence here that the other guys don't. And in this particular position, it's quite variable. They have a lot of different nucleotides at that position. And some of them are, more well-behaved. Some of them, only one taxon has a difference from another. And the ones that we are really interested in for our purposes are the ones that a couple of the organisms have one state and a couple of the other organisms have a different one. We're not so interested in these that just are unique to one organism, because they don't help us with grouping them together. But this kind of position is the kind that would be used most importantly to build a phylogeny tree, because this one implies that 1, 2, and 3 are related and 4 and 5 are related. Could be wrong, but then, and in fact is in conflict with this one that's 2 over. The one that's 2 over shows a different grouping. And I'm going to show you how we work out conflicts, but I want to be sure everybody can see how we can get homology information from the DNA just like we can from the anatomy of a skeleton or of a flower. And so here's a quick exercise. If we were working on this in a classroom, I'd have everybody build a phylogeny. But here, we'll just work through it together. Here's a data matrix that has 5 species, 5 terminal taxa, and then there are 12 of these potential markers that have already been scored. So this is not an alignment. They've already been scored, and they've even, as a hint, the one condition is the apomorphic condition, and the 0 condition is the plesiomorphic one, which in practice is told by comparing to the relatives that are outside of this group. It's called outgroup comparison. So if these 5 make a little clade, and you are looking for which of those 2 states is plesiomorphic, which is apomorphic, you would look and see which one is outside the group, implying that that was the ancestral one. But let's say, I'm just making it easier here. Let's say we know that the shared ones are sharing the homology. So can everybody see what character 1 is telling us? Assuming now I have already predetermined by my analysis that the ones are a shared derived character, right? You can see how it's at least one little bit of evidence that A and B are related to each other. And now I look at the next one, next independent marker, different bone or a different nucleotide in the genome. And I go, oh, that one is the same. It's consistent. So it's another piece of evidence. 3 is not very useful because we already hypothesize A as a separate group. So 3 doesn't really group anything. 4 doesn't group anything, but 5 does. 5 implies E, C, and D are related to each other. This one here in 6 is not very informative. These are actually called phylogenetically uninformative type of characters. But then look at this, 7, 8, 9, and 10 all show the same signal that 7, 8, 9, and 10 are related to each other. Or I mean that C and D are related to each other. And then 11 and 12 are both these kind of phylogenetically uninformative ones. So let's see if you can do tree thinking. Can everybody see how we have a tree here? Even though it's shown as Venn diagrams, we actually have a tree here. And the tree is this one. So the tree we've inferred is that A and B are sister groups, C and D are sister groups, E is related to C and D. And we had as an assumption to begin with that they're all part of one clade. So here is a phylogeny that's built from those boxes. And furthermore, you can make a hypothesis about where the characters changed. We have a hypothesis that 1 and 2 changed on that branch. See this uninformative character 3 changed there, 4 changed there. We hypothesized that 5 changed there, 6 there. And then we have this hypothesis that 7 characters changed on that branch. Okay, does that seem reasonable? There's no noise here, so it's really easy to see that this data set yields one phylogeny and the only logical one you can get from that. If you did have some, and I wanted to illustrate that this phylogenetic reasoning isn't the same as overall similarity, because if we matched up 0s and 1s without considering which one was apomorphy and which was plesiomorphy, then the diagram we would get, if we now just look across the row with A and B would certainly be the most similar to each other. They're similar in 10 of the 12 characters. And C and D are most similar to each other. They're similar also in, I believe, 10 out of the 12 characters. But the kicker is that E is more similar to A and B if you just count how many similarities there are. I think there are 7 similar, because in all of these 0s, they're similar to A and B. We're not distinguishing the difference between 1 and 0. We're just saying, are they similar? So in what's called a phenogram, it's usually is shown like this. And what it is indicating is the overall similarity of the organisms, not the phylogeny of them. And this just shows how it can go wrong, that they're not necessarily the same topology. It gets A and B and C and D right, but it misses E here. So that's a lesson that we'll come back to talk about, how you really have to analyze the data phylogenetically and not what's called phonetically or distance-wise, which is what you do here. One other thing I want to illustrate here is, what if we did have one of these conflicting characters? What if I had a character that the apparent apomorphy was shared by A and D? You see, that just doesn't work. That sort of implies this relationship, which kind of cuts across this one. That's what we call a homoplasy. And a homoplasy is a character that looked like a homology to begin with, but it turns out that it's just not once we look at it in the context of everything else. So an example of a homoplasy would be maybe the wings on a bat and the wings on a bird. They might fool you into thinking that, gosh, they're awfully similar. But then when you work out all the other anatomical and genetical characters, you find out that no, the birds and the bats are more distantly related to each other, and their ancestor didn't have wings. And so this is a independent evolution. So homoplasy is often a parallel evolution of a trait or convergent evolution or something like that. But we can still map it on the tree, even though it is incongruent. It just adds branch length in 2 places. See, so the 13 gets mapped there and there. So the 13 is mapped on 2 places. Homoplasies can't be mapped in 1 place, or else they wouldn't be homoplasies. But they can still be mapped. So our phylogeny, built on the context of all the characters, can tell us about the distribution of individual ones. And really important for today's discussion is, can you see how this is starting to get us at what the branch lengths are? Because we can reflect branch lengths in how many inferred changes there were on the branch. And that branch right under C and D looks really long. So if we were going to scale this phylogeny by the branch lengths in inferred change, this is what it would look like. It's the same topology as this. The branches are connected the same way, but you see this long branch here is reflected by all that change there. D is longer than C because there are 2 changes in D and 1 in C and so forth. So we are scaling the branch lengths by the inferred amount of change on the branch. That's one kind of a branch length that we will talk about in this in the later sections. That's what's called a phylogram, where the branch lengths are given as proportional to how many inferred changes they had. That's just one of a couple. There are 3 facets of phylodiversity, which we will talk about today. And there's really more than this, but there's, let's say, 3 important ones, which is the phylogram, which we just looked at, which is a way of representing branch lengths in terms of number of changes. Then if you can time calibrate the phylogeny and bring all the branches to the present like this, and then get the units to represent years, to represent time instead of character change, then that's what we call a chronogram. There are methods of taking, which we'll talk more about later, but you can take these data, and if you have some other evidence from fossils and so forth, you can turn the phylogram into a chronogram, which now those branch lengths represent time instead of number of character changes. Or you can, a third major way of looking at the branch lengths is to consider them all equal. So don't worry about either of these and just say, okay, all these branches are the same. And I've highlighted a PD measured on these different types of topologies, and they're different. And there's a wonderful little animation here that our colleague Matt Kling did that we had to show you. This is the California plants. So there's phylogram. There's just no, equal branch lengths, and there's time. See, the time one, everything comes to the present at the same moment. And you can just see that even though the topology of the tree is not changing, the branch lengths are changing depending on the measure that we use to describe their length. So at least these 3 facets are going to be, it's going to turn out they're all super useful for conservation. So this is one of those situations where one's not better than the other, but they are telling us different things. The mutation here is a reflection of genetic diversity. This is a reflection of amount of speciation, and this is a reflection of time. And each of those has some value in conservation decisions, and so we'll end up looking at all of them. And we thank Matt Kling for sharing this with us. Okay, so to summarize this section of the workshop, then let me just go back over and make sure we're clear on the basic concepts that the phylogeny is just the hypothesis about how the organisms are related to each other. So when they're connected at a shallow level on the tree, there's a hypothesis they're closely related to each other. If they're connected deeper on the tree, you're hypothesizing they're more distantly related to each other. So we're looking for these little pieces of shared history that we can infer from things that happened on them that maintain a signal to the present. So we're looking for these little bits of shared history. We're trying to find these markers that indicate that branch existed. And then we build up a matrix with all of those. And then we make a tree inference, which is another highly computationally intensive subject that we teach a whole semester graduate course on. So you're not going to learn how to build a phylogeny from what I just told you, but you've learned the principles of it. And then because the data sets are so big these days, you have to use computer software to help you sort out what appears to be homology and what appears to be homoplasy and so forth. But the logic is all here. One of the things we wanted to just note that's confusing to people who are not familiar with trees is that there's nothing about the orientation of these on the top that matters. They can all rotate around like one of these mobiles above a child's crib. They can all just rotate around. So D here is not more advanced than A. I could have rotated it around so that A was over here on this side. So what matters, all the organisms that are around today are around today. None of them are primitive or anything compared to others. But what does matter, as we started in one of the very first slides, was what matters is the relative recency of common ancestry. Then we have different ways of representing the branch lengths on the same exact topology without changing the connections, and those are the facets. So the facets are different ways of representing branch lengths on the same topology. So what we're going to do now is take we're going to have another discussion section, and then after that, we're going to take a short break for people to get some more coffee, and then we're going to come back together.